Hello, Dr. Raymer here, and today we're talking about sensory systems in the CNS. You can see the 3 learning outcomes for this little lesson. Today we'll focus in this video mostly on this one, and we'll leave these clinical sort of problem-solving exercises related to this work for a tutorial. So sensory information from the body enters the CNS via 2 main ascending systems. The dorsal column medial lemniscus system, or DCML, and the anterolateral system, which includes the spinal thalamic tract. So this is something people mix up quite a lot. These are 2 distinct terms. The anterolateral system is an inclusive term, which encompasses or contains multiple tracts in the spinal cord, traveling in the anterolateral white matter, including the spinal thalamic tract. Sensory information from the head and face enters the CNS via the cranial nerves. So today we're focusing on sensory inputs from the body, but just keeping in mind that information from all of these modalities is entering from the head and face via the cranial nerves, and those project to a distinct location in the thalamus, the ventroposteromedial nucleus of the thalamus, or VPM. So the dorsal column medial lemniscus pathway, or DCML, so I don't have to say the whole thing all the time, carries information about vibration, proprioception, and fine touch. And that sort of gives it away, this question then. So what tests on the neurological exam examine the function of this pathway? But you do need to make sure you review the neuro exam so that you know how those tests are performed and how to interpret those results. So for both of these sensory systems, we'll begin in the periphery at the first order neurons in the dorsal root ganglion, or DRG. Now, for the DCML system, these primary sensory neurons are the larger diameter neurons, and most of them have specialized nerve endings, such as this Pacinian corpuscle illustrated here, entering the cervical spinal cord. Now at this stage, it's worth remembering that the sensations we experience are due to differential activity of a really wide array of specialized sensory neurons in the DRG. Here we're looking at skin, glabrous and hairy skin, and you can see that there are both encapsulated nerve endings like Meissner's corpuscle and the Pacinian corpuscle. And in hairy skin, we've got nerve endings associated with the base of hairs that transduce touch and bending of the hair. Not shown in this diagram, of course, is the proprioceptors like muscle spindles. So you don't need to know all of the specifics of these different types of sensory neurons, but you do need to appreciate that there's a wide variety of sensory neurons. In the case of the DCML pathway, they're largely somewhat specialized, whereas in the case of the anterolateral system, they're mostly free nerve endings. And this is just summarized in this chart, sort of categorizing sensory neuron fiber types. And again, it's important to just have a general appreciation here of what does what. In the case of the DCML system, we're looking at this top category, type 1 and type 2 sensory neurons. They're larger in diameter, they're myelinated, and they tend to have specialized end organs for receptors. Conversely, the anterolateral system, conveying information about pain and temperature and itch, is type 3 and 4 sensory neurons. They're smaller, they're not all myelinated. C fibers, of course, are unmyelinated, and most of them have bare nerve endings or free nerve endings rather than specialized end organs such as muscle spindles or structures in the skin. Now, while I go on this little tangent away from the pathways, I just want to remind us about a few things that are really important when we think about neuroanatomy of the sensory systems. The first reminder is that spinal nerves transmitting information between the spinal cord and the body are mixed. That is, they contain both motor and sensory fibers. And we tend to forget this because we think about motor function and sensation separately. Of course, we know they're functionally linked and integrated in the CNS, but actually motor and sensory fibers are traveling together in peripheral nerves in the PNS. And that intermingling occurs really proximal to the spinal cord. So if you look at this diagram up here, the kind of diagram that we're all used to seeing, dorsal roots with information entering the cord and ventral roots with information exiting. If we look at this box section in enlargement, it's a great reminder that immediately proximal to that dorsal root ganglion, immediately distal to that dorsal root ganglion, right here, where the spinal nerve arises, at that stage, it's a mixed nerve. So it contains motor fibers coming from the ventral root and sensory fibers en route to the dorsal root ganglion. And just as a final reminder before we go on, we know that each segment of the spinal cord and each corresponding spinal nerve and dorsal root ganglion receive sensory information from a specific dermatome or patch of skin. The dermatomes are sort of segmentally organized, and it's really easy to appreciate this when we look at dermatomes on the torso because they have this predictable banded pattern. Two sort of important ones that people often use as landmarks. T4 is about at the level of the nipples, and T10 is at the level of the umbilicus. So those are just good landmarks. There are mnemonics for them. T4 is teat por, which is a bit weird, but anyway, teat por, nipple. And T10, button, so belly button. So some fun mnemonics for you to use. The banded pattern that we see on the torso kind of falls apart when we look at the limbs. So you can see that in the limbs, all of a sudden, this person's standing with their arm to the side in some external rotation. And when you look at the axilla, the axillary region, all of a sudden C5 is opposed to T1. And to see how this happens, we have to look at embryonic development. So in embryonic development, of course, we have this nicely segmented embryo, and then out of it develops little limb buds. That happens about 4 weeks of embryonic development. They start to grow outward, and as they do, there's some leading dermatomes, just the leading skin, and they kind of get pinched off from the rest of the limb. So the most distal dermatomes aren't where you would expect them to be. That's just of the way limb growth progresses in development. And so this is what produces this pattern. And you can see it here. It's easier to envision what happens with the lower limbs if you think of the legs as being splayed out sideways in a horizontal split. But that's how the dermatomes arise. You'll also see that dermatome maps don't always agree. You'll see variability in the maps, and that's accurate. So different people have different specific dermatome maps. Just like everything else, this is an idealized dermatome map. In addition, the dermatomes sometimes overlap, so they're not perfectly delineated. Now, with all that in mind, let's get back to the DCML. So here we are, we're outside the spinal cord, it happens to be the cervical spinal cord, and we got information from a specialized receptor flowing toward the dorsal root ganglion. It's a Pacinian corpuscle, I think, by this, so perhaps it's a vibration or a deep touch, something like that. And that information flows via the dorsal root ganglion into the dorsal horn of the spinal cord. Now, once in the dorsal horn of the spinal cord, you'll see that some central processes of this pathway do give off little collateral branches here. And so that's what's shown in this diagram. There are some collateral branches. Those largely mediate reflexes, spinal reflexes. Dogmatically, though, most of those central processes continue without synapsing. So they ascend in the dorsal columns and join one of these two tracts, the fasciculus gracilis or the fasciculus cuneatus. Because we're in the cervical spinal cord here, we know that this information is going to travel in the fasciculus cuneatus. So cuneatus, cervical, they both start with C. The fasciculus cuneatus is also located lateral to the fasciculus gracilis. Now, the second order neurons in this pathway are located here in the caudal medulla. And since we're traveling in the fasciculus cuneatus in this instance, we're going to synapse on the nucleus cuneatus. And the nucleus cuneatus neuron will be the second order neuron here. we'll move to this larger image. Here's my nucleus cuneatus. And you can see that the nucleus gracilis is the gray nucleus immediately medial to it in the caudal medulla. Now from here, the nucleus cuneatus neuron is going to extend an axon across the midline. And as it crosses, that sort of tract of crossing fibers in the caudal medulla is called the internal arcuate fibers. So this is the point of decussation of the pathway, and while crossing, they're called internal arcuate fibers. After crossing on the other side of the medulla, the fibers coalesce and they form the medial lemniscus. And the medial lemniscus then ascends all the way to the thalamus. So just important to remember what's called what where. It's the dorsal columns prior to that synapse in the nucleus cuneatus, the internal arcuate fibers while they cross the midline, and the medial lemniscus traveling through the whole brainstem all the way to the thalamus. So once at the level of the thalamus, the medial lemniscus synapses on tertiary or third order sensory neurons. And those are located, of course, in the ventral posterior lateral nucleus of the thalamus, or the VPL thalamus. From there, those tertiary neurons project to the ipsilateral primary somatosensory cortex. Now, at this stage, it's worth reminding ourselves that although we typically describe the thalamus as a deep brain nucleus, it's actually a big bundle of specialized nuclei, and that includes the VPL and the VPM, which mediate somatosensation from the body and the face and head, respectively. So all of these nuclei are named. They have sensible names, like VPL is ventral posterior lateral, which is just where it happens to be in the thalamus. And then they have some specific names, like the pulvinar nucleus, for example. Now, we often describe thalamic nuclei as being relay nuclei. That is, they receive input and project to cortex. In fact, they're also in reciprocal communication with functionally related bits of cortex. So in the case we're talking about for somatosensory cortex, it's obviously in communication with the VPL thalamus, but note that these arrows are bidirectional. So the VPL thalamus is conveying input to somatosensory cortex and vice versa. And of course, here's the information from the face that impinges upon the VPM thalamus and in turn is in bidirectional communication with somatosensory cortex. At the same time, it's not just about sensation, right? The motor and premotor cortex communicate with the ventral lateral thalamus. The prefrontal cortex communicates with mediodorsal thalamus, and so on. So there's a whole set of reciprocal connections linking thalamic nuclei to cortex, subserving a whole huge range of function. Now, these reciprocal connections linking the thalamus to the other parts of the brain are broadly described as thalamic radiations. And if we return to the internal capsule for a minute, it's really important to remember that these thalamic radiations are also contained within it. So here we've got our white matter, the internal capsule, that characteristic V, and we know we've got some somatotopy of fibers organized in the internal capsule. We've talked about this in the context of motor function. So motor fibers governing the face and head in the corticobulbar tract are here near the genu, while motor fibers in the corticospinal tract governing the body are sort of organized from rostral to caudal bits of body, anterior to posterior, within that posterior limb of the internal capsule. So there's some somatotopy here. But before you get distracted and think only about motor, it's important to remember these thalamic radiations. So thalamic radiations, including the somatosensory radiation, the connections between thalamus and somatosensory cortex, are also found in the posterior limb of the internal capsule, as are portions of the optic radiations, for example, traveling from the lateral geniculate nucleus to the primary visual cortex. So these thalamic radiations can also be damaged in a lacunar internal capsule stroke, and that creates the pattern of deficits. Now, when you see everything that's going on anatomically in the internal capsule, you begin to appreciate why such a small internal capsule stroke has the ability to produce the same signs and symptoms as a relatively large cortical stroke. And the difference, of course, is cortical sign involvement. So cortical signs are things that can help you localize damage to cortex. Aphasia, apraxia, neglect, extinction, these are all examples of cortical signs. Others include visual field deficits, like loss of half or a quadrant of the visual field, executive function or personality changes, and then some really interesting specific deficits, like agraphia. So you lose the ability to write. All of your other language function might be intact, but agraphia is specifically loss of ability to write. Alexia, loss of ability to read. Acalculia, loss of the ability to work with numbers specifically, and I don't mean kind of crap at doing math in your head. I mean, if given two numbers, you might not be able to tell which number is larger than the other. And agnosia, which is loss of ability to name things. And this can take all different forms, but an example of somatosensory agnosia would be if someone put a key in your hand, and although without looking you could feel all the features of the key and describe it, you would not be able to name it as a key. When you look at it, you might be able to instantly say key. That's an example of an agnosia. And all of these are signs that indicate a large vessel stroke over a small lacunar infarct that's located subcortically. So after that segue into thalamic and internal capsule anatomy, let's return to our sensory systems and talk about the other major system, the intralateral system. So we know that the system carries information about crude touch, pain, and temperature, and I'll add itch to this list. And it contains multiple tracts. So all of these tracts travel in the anterolateral white matter of the spinal cord. All of them receive information from small diameter primary afferents or primary sensory neurons in the dorsal ganglion, but all travel to different parts of the brain. So this schematic sort of shows the anterolateral system all together, which I think is a nice way to look at it, because the separate tracts are just named for where they project to. You can see in the schematic that S1 here is the primary somatosensory cortex, but you could also see that information from this pathway is transmitted to all kinds of other parts of the brain. So here's S2, somatosensory association cortex, for example. The prefrontal cortex receives information from the pathway, the hypothalamus, many brainstem nuclei like the reticular formation, the amygdala. All of these parts of the brain and others receive information, particularly about pain, right? So the emotional aspects of pain and the contextual aspects of pain, those aren't mediated by S1, by the primary somatosensory cortex, but by other parts of the brain. So the spinal thalamic tract is the one we know and love, and it is the one we clinically test on the neuro exam. So this mediates discriminative aspects of pain and temperature sensation, such as where on my body is this happening and how intense is the pain or the temperature change. The spinal thalamic tract, neurons in that tract, as the name would suggest, synapse in VPL thalamus, but on distinct tertiary neurons from the DCML. So it's not the same neurons in VPL thalamus receiving input from all modalities. The spinal reticular tract, it does something different. So it conveys emotional and arousal aspects of pain. And as the name would suggest here, one of the primary sites of termination of the spinal reticular tract is the reticular formation in the brainstem. So these nuclei are found at multiple levels in the brainstem and critically mediate arousal. These neurons project to distinct, these systems project to distinct thalamic nuclei. So that's an important difference between the spinal thalamic tract and something like the spinal reticular tract in this system. They're not using the same nuclei in the thalamus. And the spinal mesencephalic tract is another tract. it projects, it's not shown here, I don't think, to the midbrain periaqueductal gray matter. And so it would project maybe about here, unless they're trying to show it here. The periaqueductal gray in the midline is critical for modulation of pain. And so these are 3 pathways, and there are other named pathways in this system. Now this slide contains the same written information as the previous slide, but it's just to show you on this different schematic where these pathways are represented. So axons in the spinal reticular tract, for example, would exit the interlateral columns in the brainstem to synapson reticular formation nuclei. Similarly, in the midbrain, this is where that spinal mesocephalic tract exits to synapse in the periaqueductal gray, and so on. So just getting the idea that all of these axons travel together in the interlateral white matter, but they exit to different parts of the brain. Now, just like for the dorsal column medial meniscus pathway, it's important to know how you test the function of the interlateral system on the neuroexam. If we look at first order neurons this time in the interlateral system, most first order neurons have free nerve endings or bare nerve endings. They're smaller in diameter, and many of them are unmyelinated fibers. When you see this information entering the spinal cord, so this time it might be temperature information, for example, again it enters by way of the dorsal root ganglion, and the central process of that primary afferent enters the dorsal horn. Now, we typically see it represented like this, synapsing in the dorsal horn, and that's sort of it. What actually happens is a little more complicated. So these axons often ascend or descend for a couple of segments before synapsing, and that is what creates Lissauer's tract. So this is shown on this schematic. Here we have an A delta fiber or a C fiber, so a couple of axons, primary afferents that would contribute to the interlateral system, and you can see them branching. And so what they typically do is enter, and they branch rostrally or caudally to travel down 1 or 2 segments before synapsing in the dorsal horn of the gray matter. And if you have trouble visualizing this little 3-minute video does a pretty good job of showing you what Lissauer's tract or the posterolateral tract is. Just those little uppy-downy movements of the axons prior to synapsing in the dorsal horn. So the second order neurons for the interlateral system are of course located in the dorsal horn, and they're located in named nuclei. So the nucleus proprius is here, the substantia gelatinosa is here, and recall that these are laminar areas. The Rexed's laminae were based on cytoarchitecture, like Brodmann's areas of the brain, but these nuclei are based on function. And so if you hear these terms, they're just the portion, a column of gray matter that contain the second order neurons of the interlateral system. Now from there, the second order neurons, I'll trace the blue one, so I'm not going red on blue, extend axons that decussate in the anterior white commissure of the spinal cord. So this little bit of white matter is the anterior white commissure, and that is the crossing point for the interlateral system. Then those axons enter the interlateral white matter and ascend. So this is the spinal thalamic tract. We're talking about this neuron extending to the thalamus. Now, of course, the third order or tertiary sensory neurons extend again from the VPL thalamus. That's where they're located. They receive input from the spinal thalamic tract, the one we're talking about in this case, and that in turn ascends ipsilaterally to the primary somatosensory cortex. You can see here information that's headed to another pathway, another interlateral system pathway is impinging on distinct thalamic nuclei, so not VPL thalamus. And from those nuclei, for example, it might go to different areas in the brain, like somatosensory association cortex, or other cortices, other parts of the brain that give emotional context to the pain, for example. Now, just a few more things that's important to keep in mind before we start linking structure to function and mapping lesion deficits is that somatotopy persists in these somatosensory tracts within the spinal cord. So if we look at the dorsal columns, for the dorsal column portion of the dorsal column medial lemniscus tract, we can see that within those columns, caudal to cranial maps roughly medial to lateral. If we look at the anterolateral system, we can see another pattern, and that's that caudal to cranial maps roughly ventral to dorsal. So now we've layered on sensory function and sensory neuroanatomy onto our motor neuroanatomy. And it's really these three long tracts in the nervous system in combination with some cortical signs that help us localize lesions within the CNS. Remember this critical pattern: rostral to the decussation, you get symptoms on the contralateral side; caudal to the decussation, symptoms on the ipsilateral side. And that's sort of the mantra you need when you're linking damage to functional outcome. Now, in terms of sensory deficits specifically, it's really important to understand just a few clinically relevant things. We tend to think of damage to a sensory pathway producing loss of sensation. So that's the simplest scenario. In reality, it can also produce abnormal sensation, and the general term for this is paresthesia. So like loss of sensation, this abnormal sensation is typically present caudal to the level of injury in predictable ipsilateral, contralateral ways. And it's not just the location of the abnormal sensation, but the type of abnormal sensation can have value in localizing lesions if damage is occurring to one pathway or another. So dorsal column medial lemniscus damage, if it doesn't produce loss of predictable modalities, can produce tingling or numbness or tightness. One description is gauze on the fingers, like wrapped tight. Anterolateral system damage, in contrast, if it doesn't produce loss of predictable modalities, can induce sharp or tingling or burning pain. And so these paresthesias can also be used to localize damage. A few clinical terms in addition to paresthesia, which is abnormal sensation. Dysesthesia is unpleasant abnormal sensation, so painful or uncomfortable paresthesia. Allodynia is pain in response to a normally innocuous stimuli. So allodynia would be pain in response to wind on your face or a light brushing on your arm or clothing present on your skin. Hyperalgesia, in contrast, is an exaggerated or prolonged pain response in response to already painful stimuli. So a pinprick could produce really prolonged pain. That's an example of hyperalgesia. So just a few clinical things to keep in mind before we start linking anatomy to clinical outcome with some cases.